Yeah, I did. Yeah. Okay, no, brilliant. All right. Yeah. All right, okay, good afternoon, everybody. Thank you for being here today amidst the cold, wet wind and also technical difficulties. Today, we are really excited for this talk that's being given by Professor Simon Schultz. He's a professor of neurotechnology over at Imperial, who we work with quite closely. And yeah, highly anticipated. Can't wait to hear what he has to say. So please take it away.
Thank you very much. What I've decided to do, I give you a sort of slightly focused talk on some of the recent work we've been doing in my lab on using light to probe brain function. Before I do that, however, maybe I should, just sort of verbally tell you a little bit about the Neurotechnology Center at Imperial and sort of how it emerged. We've been around since sort of about 2008 or something like 2007, 2008, something like that. Coming up within the Faculty of Engineering at Imperial. So I was employed at Imperial as a lecturer in 2004, and I recall the Dean of Engineering coming around and basically, we all presented our research, and she sort of said, where's the engineering in what you're doing? And I said, well, basically I'm doing engineering, making new tools to do neuroscience, right? And so we sort of, thank you very much. That's great. We realized at that point we needed to sell it better. And so searched around a bit and came up with this word neurotechnology. Basically it sort of combined neuroscience and engineering in what we thought seemed the right way. The only other people using the word at the time were MIT, that had the MIT, the MINT program, McGovern Institute of Neurotechnology. And there are a few centers around the world were doing what they call neural engineering, which is pretty much, which could be the same thing, depending on how you look at it. Maybe I would use the word neurotechnology to be a little bit broader and span some things like, for instance, molecular neurotechnology, new sort of molecular biophysics, new probes, that kind of thing would definitely be neurotechnology. And maybe you wouldn't find it in a center that calls themselves a neural engineering center. But largely I think that's sort of synonymous. So we originally applied to there was this sort of round to apply for strategic funding, and I applied for a small grant to set up a sort of a ground roots initiative Imperial called the Strategic Initiative in Neurotechnology. So I just wanted to persuade the Dean of Engineering to fund SIN, S-I-N. Right? But anyway, and it came from there. We had a lot of interest from different departments in engineering, but also the medical school, the Faculty of Natural Sciences, as they are now. Basically sort of around sort of 60 sort of lab heads from across Imperial College sort of got involved, and we then set up we set up a stream of our masters. We had an MSc in biomedical engineering, so I added a neurotechnology stream of that, and that was really the first sort of taught program we had in neurotechnology. And we for that, you want to introduce a new master's, you need to sort of not just rebadge other courses, you got to introduce a few new courses for it. So we introduced, I had already introduced a computational neuroscience course. We added to it a brain machine interfaces course. And those are still being taught, not by myself, unfortunately. I went away on sabbatical and had to hand them on to someone else, unfortunately. But anyway, they're still being taught. And that then led into a master's where we created a master's by research and used that basically to bid for funds from the UK government, Engineering and Physical Sciences Research Council, and from a bunch of companies, including Medtronic, a big instrument manufacturer, GSK, so the big pharma company. They had this initiative that actually later was spun out to become the company Galvani Bioelectronics, that basically do sort of peripheral nerve stimulation technology. McLaren, actually you think of McLaren as the Formula One. McLaren Formula One, they actually have McLaren Applied Technology as a sort of sister company, which basically tries to find biomedical applications of some of the sensor technology they developed for Formula One. So they funded some studentships with us, and a bunch of other companies were involved as well. So we put that together. We had a bid for £10 million centre for neurotechnology based around this doctoral training centre, which is sort of trained around 60 PhD students over its duration. Unfortunately, that's come to an end. The funding was 2014 to 22, basically, and the UK government, they had these doctoral training sort of schemes, but they reduced the amount of funding for it sort of over time. So unfortunately, we don't have funding for that, but we have still a lot of research going on associated with the centre. The centre still exists as a sort of vessel for sort of cross-faculty sort of collaboration, and we have other sort of big research grants that collaborative research grants that are involved there. So that just gives you a bit of context. And the other thing I want to say, what's been very interesting over the last few years is the emergence of some groups like you, so student interest groups interested in neurotechnology. I would never have predicted that would have happened 15 years ago. And then suddenly there is this splurge, particularly in the UK. I think it's not, I mean, there are in other countries too, but not to the same extent for some reason. It's been, I think, quite a thing here. And so I think that's great. And anyway, hopefully you'll be the next generation of neurotechnology researchers, or neurotechnology users, or neurotechnology advocates, or whatever. Okay. So okay. So the title of my talk is Watching Thought, Using Light and Viruses to Probe Brain Function and Dysfunction. This is kind of a gratuitous picture, but it is actually a confocal microscopy picture from my lab, which I think we should re-encounter later on, so I'll explain the thing. So I'm going to be talking to you about optical neurotechnology, and it's not the only type of neurotechnology that my group have worked on, but it's kind of one of our main focuses, and probably is different to what you might hear from some of the other speakers. So I can sort of focus on that. So optical neurotechnology to be using light to stimulate the brain, and here is an example of optogenetics. So you might have come across this channel rhodopsin. Basically, you can genetically modify neurons to basically sort of make them light activatable, or even sort of light suppressible. And here's examples of channel rhodopsin, halo rhodopsin, ARCH, sort of light activated proton pump. And we've used a few of these. So it can be used to stimulate brain function, and it can also be used to probe or read or measure brain activity as well. And we've done both of these sides of things in my group. I mean, I'm mostly talking about the reading out today, but we have done a bit of work on stimulation. I can maybe tell you a little bit about the end. And there are many sort of modalities for doing that. One approach I'll maybe sort of say a little bit about is single photon fluorescence imaging. So basically, fluorescent proteins are a sort of a wonderful thing. You can modify biological systems to express them, and then you can start to see structure, but you can also make build them up into biosensors that can tell you things about the chemical environment. So example being the many calcium sort of biosensors. GCAMP is one that we've used quite a lot, a sort of genetically encodable calcium sensor. Once you've got that into a neural circuit, then the fluorescence will depend upon the local calcium concentration. Of course, as you know, when neurons fire action potentials, there's an influx of calcium, so you can use it indirectly as a readout of neural activity. There are other more direct readouts of neural activity. There are voltage sensors, for instance, that we've worked on. Their signal-to-noise ratio is still sort of orders of magnitude lower, so that the calcium sensors still give you the best sort of bang for bucks, so to speak. Now, how do you use them? So this is an example using single photon fluorescence. That's and I think I've got a slide on fluorescence in a minute, so I'll leave it to them. But basically, you can have a wide field microscope. You excite, if you want to have fluorescence, you want to basically have some light coming in, which causes excitation, and then you see the light given off, and you can basically record it with a camera. Now that can be miniaturized now, so you can have these little mini scopes, so you can have that. Oh, sorry, I think the live stream is not working.
No, it's fine. Keep
Okay,
going.
good. Keep going. Yeah. So basically, you can have a little head-mounted mini scope, and on a mouse or a rat, and they weigh sort of a couple of grams. So it's sort of the limit of what you can put on a mouse, but it can be done. And then they can sort of move around the environment and do sort of behavioral tasks, etc. And you can image the brain while that's happening. So that's great, and I'll tell you about going in that direction and some sort of slightly improved technology there. At the other end of things, it can be used, light can be used to probe brain function in humans as well. And this is a device, this is basically called functional near infrared spectroscopy. It sort of measures, again, an indirect measure of neural activity through the bold signal, or something very similar to the bold signal, just like in fMRI. The difference is you don't need to take a person in. Well, there's a few differences. There's pluses and minuses. The plus is you don't need to take the person and put them in a scanner and sort of have them lie down, etc. You can basically have them walk around, do things. On the downside, the spatial resolution is much more limited. The temporal resolution can actually be slightly better for various reasons with the latest FNIS technology. So I won't talk about it today, but we actually have a little spin-out company called Connectome Health that basically we've got a pilot study called the Lucid study, where we're basically having people do cognitive tasks, monitoring their brain, and over time getting them sort of come back, etc., and trying to use it or develop a tool to track brain health. But the approach that I want to sort of talk to you about today is what I call precision circuit neuroscience. So the idea is, so obviously I showed you on a slide before doing something in a mouse. So we can use small animal models of neural circuit disorders, and there are a lot of them. They vary in how good they are, and they should be normally taken as models of a specific aspect of the disorder. Example, there are mouse models of Alzheimer's disease. The mouse does not have Alzheimer's disease, but you might, for instance, have some human some genes sort of knocked in that are from human families with familial early onset Alzheimer's disease that have mutations in particular proteins that cause the production of amyloid plaques. And then you can start to look at what is the effect of amyloid plaques on tissue responses, for instance. So that's sort of an example. We can use small animal models of neural circuit disorders, and then use optical tools to study them. So we can modify circuits by optical genetics. We can stimulate and see what the response is. We can read out using single photon or 2-photon calcium imaging. I'm going to be talking about that today. But also we can use electrophysiological approaches, in vivo multi-electric array recording, like NeuroPixels devices that have sort of many hundreds of electrodes, or the other extreme, if you want to have only one or a small number of cells, but you want the best possible signals, you can use patch clamp electrophysiology. And something that we've worked on is basically doing 2-photon targeted patch clamp electrophysiologies, that we can target patch clamp pipettes onto cells based upon their sort of genetic characteristics. We might label a particular type of cell, say, example being a GAD67 positive interneuron. So a particular type of interneuron in the brain, we can then make those glow and then steam the pipettes down just to record from those. So this is all things that we can do in animal models. So it's great technology, but you're never going to be able to do it in a human. And then the idea is, if you want to then, let's say you've done something really useful here, you've made an effective therapeutic for a disease condition. How do you translate it to human? The idea is to replace these interventions. So you use the most precise approach in the animal model to understand it best, and then you replace these interventions with things that can be used in humans. So for the stimulation, we have access to things like deep brain stimulation, transcranial electrical and magnetic stimulation, example being temporal interference stimulation, one of the recent techniques. And particularly focused ultrasound is something that over the past year or two has had a massive amount of attention, and I think is probably going to be one of the main ways we can translate things like optogenetic stimulation into or ideas that have come from things like optogenetic stimulation into things that can be used in a human. So a lot of ongoing work in that area. And then for the readout aspect, again, we can't really do 2-photon calcium imaging, for instance, in a human. But we can do other things. And it depends on what you want to look at. If what you care about are your brain rhythms during sleep, then electroencephalography is probably a good way to do it. I've talked about FNIRS already. Another thing very useful is magnetoencephalography, where you can get effectively a whole brain or near whole brain, very high temporal resolution sort of readout. And so we're using that to look, for instance, at particular sort of high frequency oscillations with sharp wave ripples that are generated by the hippocampus. And at the moment, we've got an ongoing effort to try and detect them using magnetoencephalography. And a collaboration with the group in Japan have got a paper demonstrating that you can do this. We're now sort of trying to robustly replicate it. Okay, so that's just an introduction on the sort of the general approach to science we're looking at. This is my third university, Oxford. You've probably come across this sort of through the looking glass. So I often use this slide as sort of an intro to talks. I came across. Okay. Sorry. Yeah. So I've given you this. So basically, Alice in Wonderland. I'm sure you all know the story of Alice in Wonderland. I've given this talk in different countries because in some countries they've not heard of Alice in Wonderland, so I have to explain it. but basically, so Alice goes through a looking glass, which in her case was a mirror, and finds on the other side of it this sort of weird and wonderful world with talking rabbits and all of this kind of thing, right? Now we kind of are going through a looking glass as well. Our weird and wonderful world is the brain, and there's lots of sort of interesting things in there. And the reason I show this is mostly because I live near Guildford, and I went running and I found actually what used to be Lewis Carroll's home. So Lewis Carroll wrote these books in Oxford, actually in the garden of Christ Church College in Oxford. But he lived his permanent home was in Guildford, and this is like the location where he used to live. It's actually a park now. And there's this nice statue of Alice sort of going through the looking glass. So that's Alice's looking glass. It was a mirror. What does our looking glass look like? It's actually more like this. Maybe not as pretty. And particularly this. Okay, so this is a microscope. Actually, we call it a mesoscope just because it sounds better than microscope. Basically, we can image much bigger sort of areas of brain. This is the objective lens. For comparison, okay, so this is about as big as your head, okay, maybe a bit bigger. You would normally think of objective lenses being about so big, right? Can we see another one on that? No, anyway, you can see the size of screwdrivers and things there, right? So basically, with a 2-photon microscope, which is the type of microscope that we're mostly using, I'll explain in a minute. Traditionally, you had a field of view of about 500 microns. Okay, so for that field of view, you can maybe look at say 300 neurons simultaneously. Okay, in recent years, they've sort of expanded around a millimeter. The standard one view by now maybe is about a millimeter field of view, and that's it's hundreds of neurons. This has got a 5 millimeter field of view, so we can image basically a 5 by 5 millimeter area. Within that, we typically would image, say, at most 5 by 3 millimeters, because there's a trade-off of the speed, the frame rate which we can image at, and the area and the signal to noise ratio and all of these things. So we limit it a bit more, but we can image anywhere within that 5 millimeters. Now, a mouse brain is about 11 millimeters in length. So we're sort of getting to the scale of a mouse brain. I'm going to show you a microscope later where we have an 8 millimeter field of view. So we've got a newer microscope, but this is one built in my lab. The other one we've used elsewhere. And this is the objective. So this is our looking glass. This objective lens is custom made objective lens as a company in California, that fabricated it for us. Very hard to find companies, optics companies, that want to make something like this, because it's, you know, it's very expensive and they don't get too many orders. It cost us $200,000 US to have it made. And then unfortunately, this was just after the pandemic, and there was basically these big sort of supply chain runs, and I think there was some issue of, firstly, the UK currency collapsed. That was one bad thing, and then secondly, I think the glass. I think maybe the Taiwan semiconductor companies were using all of a particular type of glass, and you couldn't get that glass at all, and you needed to make these things. So basically, the company came back and said, "Oh no, we're going to charge you an extra $50,000." Grants don't work like that. So we got together with a bunch of other labs. We had like 3 or 4 other labs that were interested in building one of these lenses as well, and all sort of negotiated together and managed to get them to bring it back down in this condition. So we got them making it. It was a interesting experience. Okay, so this is the microscope. You'll see some examples of this later. This little airbed, this thing that's resting on here, this is actually, you probably can't quite see from where you are, but there's a lot of little dots there. They're little holes. Yeah. So,
when you say 5mm
Yeah.
view,
Yeah.
is a bit out of this top? Is it all in focus? Like it's like you can see everything that's in the field of view, or
yeah, you'll see some examples. Yes. Yeah,
Right.
exactly. Now to get that really working perfectly, you need adaptive optics. We don't have adaptive optics on this one. We should still see, pretty much the field of view, but it could be improved with adaptive optics. The other one, the 8mm one, we do have, and that's necessary. Right? Once you start getting to really big fields of view, it's really necessary to sort of compensate differently across the field of view for scattering and various things. So yeah, okay. So basically, this is this is an airbed, and we have a little carbon fiber cage. So this is where the mouse is head fixed. So we implant an imaging chamber and a head post surgically into the mouse, and we do a viral injection into the hippocampus. We'll see that in a minute. Mouse is head fixed here, and then it runs on a little floating platform here. So basically, well, anyway, we'll see that in just a minute. So we can actually use this to do experiments in behaving mice. Now, how do we actually see what's going on? So this is I've described to you before and shown you an image of single photon fluorescence. So that's what we're seeing over here. So basically, we've got a fluorophore. Yeah, everything's a fluorophore to some probability, but things like GFP are much more efficient ones. We have some light that comes in, like a photon comes in, causes an electron to go up in energy level from the ground state to this excited state. It relaxes back down sort of short time later and emits another photon with a wavelength just a little bit longer. So it obviously it loses a little bit of energy in this process. So the wavelength of the light emitted is just a little bit longer than the excited wavelength. Now that's all very well, but it doesn't penetrate that deep through tissue. We'll see that in a minute. See it now. Okay, so this is an example of this kind of fluorescence. So this is basically a vial with a fluorescent dye, and this is our single photon fluorescence here. And this is the plane of focus. And basically you can see that, so first thing you can see is that actually the point spread function of the light as you go away from the focus is, you can pick up light from a long way away from the focal plane. And so it's not very tightly bound. And that has one particular consequence, or two consequences. One is that the light would tend to be a bit sort of smeared out. And then the second is you can't penetrate very deep through tissue. So you can really image only a few hundred microns deep using this kind of approach. Now, the alternative is two photon fluorescence. So for this, you basically you simultaneously absorb a pair of photons to cause that electron to go up the energy level. The photon given off when it relaxes down is basically like the same as this one here. So that means we need to provide excitation light, which has got half the energy, and but basically each of these photons has got half the energy, right? And therefore double the wavelength. So we need around twice the wavelength there. So that's going to be basically near infrared light. Now, the downside of this is these events are much rarer, right? For 2 photons to be simultaneously absorbed within around 10 to the minus 15 seconds, within around 100 femtoseconds is what you need, then that's rarer. So the way to achieve it is to use a pulse laser. So the laser might be around, say, 2.5 watts average power, but basically you have 100 femtosecond long pulses, which are repeated at a rate of about 80 megahertz. And when the pulse is on, you've got a very sort of high intensity light. So there's a high probability of this occurring for a very brief period of time. But then the pulse goes down so that you don't have your average power you're going into the tissue is not too high, because if you just kept that going, you cook the tissue very quickly. So by pulsing this, we can have this 2 photon excitation. And the nice thing about 2 photon excitation is, so there's a few good things. Okay, firstly, that point spread function, that point, like that's the focal plane. So if you see a photon, you know exactly where it came from. You know, it came from that spot. We can scan that spot around in 3D. It's smaller than individual neurons. So if we've got our scanning point in a neuron, we know that photon came from that neuron. So we can use that as a way to read that out. But of course, the fact that we've got to scan it around is going to slow things down a little bit. But we can scan fast. Now the second thing is, because of the physics of this process, actually it turns out that you can penetrate much deeper through tissue. So you can image basically around 500 or 600 microns deep through tissue using this approach. And we can see an example of that right here. So this is one from my lab from quite a long time ago now. These are layer 5 pyramidal cells, so they're around sort of 700 microns deep. What we've done here is we've imaged a plane at a time, and now I'm reconstructing into a sort of a volume that I'm then rotating around and intensity projection. You can see there. So that's using green fluorescent protein. And so using 2 photon, we can basically see, down to layer 5 to that approach. Using functional indicators like GCamp, we can probably see down to about there. Like they're not quite as good. GFP is better, basically, to be able to image deeper. Now, that's 2 photon fluorescence. There's something else we can do as well called 3 photon fluorescence. We've actually got a 3 photon microscope over Imperial, and using that we can see around 1.2 millimeters or 1.3 millimeters through tissue as well. But with some caveats, it's a little slower to scan this. So, okay, those are our tools. So we're going to be using 2 photon fluorescence and using 2 photon microscopes, or in some cases 2 photon mesoscopes, to basically image neural circuits while mice are doing things. What kind of questions are we looking at? So the main focus is on memory disorders, and sort of how they basically affect Alzheimer's disease, or how they're impacted in Alzheimer's disease. Basically, Alzheimer's disease is the most common form of dementia. The statistics are with dementia overall, which is now the leading cause of death since 2015. The small blip during the pandemic. It's been the leading cause of death in the UK. That's also true in probably most other Western countries now, and sort of increasingly a sort of a worldwide phenomenon. It's around 7th worldwide. So it's leading cause of death, and of course, as you know, it's something that the greatest cost comes, not from death per se, but actually from the long period sort of leading up to that. So it's got sort of massive social and economic cost. A quarter of all hospital beds are occupied by people living with dementia. So it's a big problem to solve. And we're interested in looking at it from the brain circuitry side of things. There'll be a lot of important aspects of it. What happens to brain circuitry during the progression of Alzheimer's disease? Well, the first areas damaged are the hippocampus and connected structures such as the entorhinal cortex. And you have 2 things going on. So basically, you have amyloid and tau pathology. So you've got these extracellular amyloid plaques and these intracellular sort of tau tangles that both occur, and they occur as part of a sort of a complex sort of biochemical cascade. Possibly Alzheimer's, so possibly amyloid is a little bit earlier in that cascade than tau, but they are both sort of nested in it. So it's sort of quite complex. The key thing is, okay, sort of coming down here, you've got basically these sort of APP mutations and various things end up, resulting in amyloid beta, tau. Amyloid beta may have a functional role in the brain. There's a lot of interesting things coming out in the last few years on how actually it may it may be a defense mechanism against viral infection in the brain, for instance. This is still being sort of settled out, but it probably it's there for a reason for sure. But you have these sort of out of control sort of chain reactions involving amyloid beta that result in synaptic dysfunction. So cells start losing synapses. But it doesn't stop there. Cells lose synapses, but actually the circuits react to that. And in the end, you have changes in local circuit function. And in particular, there's something called aberrant excitability that sort of happens. So if you have amyloid beta, you have basically some neurons showing hyperactivity and some neurons showing hypoactivity. And then you have sort of compensatory mechanisms in the circuit, which are maybe sort of adjusting for that. So there's this whole sort of network dysfunction. So we were interested in looking at, well, actually, what is happening in this network dysfunction that sort of, between amyloid and basically loss of information processing and memory capacity. So do that. We've been doing experiments in mice. We use the 5xFAD mouse model. Here we've been using two different mouse models. So the 5xFAD model is basically a model with 5 mutations from basically human familial Alzheimer's disease to the APP pathway. Now we take those mice, because we're using mouse models of Alzheimer's disease or mouse models of any disease really, it makes it a bit more difficult to use transgenic mouse lines. So there are transgenic mouse lines that you can get, which have, for instance, in pyramidal cells, they may have GCAM6. So they don't tend to have the latest calcium indicators, but they have some calcium indicators. But if you want to combine those with the disease models, you've got to do this sort of intersectional breeding program where you use huge numbers of mice in the colony to produce the few that you need for the experiment. It becomes inordinately expensive. So the approach we've taken for this, by and large, has been to use viral injection instead. And with viral injection, firstly, the mouse is normal up until the point that you inject the virus. So you don't have like this sort of developmental compensation issues. You can use the latest and greatest sort of calcium, genetically encodable calcium indicators, for instance, which are evolving every year. And we're now on GCAM8 is the current one we're using. When we started these projects, we were using GCAM6. We moved to GCAM7. Most of the data in our paper we've just submitted is GCAM7, and the newer ones are with GCAM8. So what we do now, I've sort of mentioned that we can only image about 500 or 600 microns deep with 2-photon microscopy. Hippocampus is deeper than that. Okay, so what we have to do is we have to aspirate a little bit of the overlying cortex, implant a cannula with a little glass window down. We aspirate down to the level of the corpus callosum, which sort of provides a good stopping point in the mouse. And we implant this sort of cannula imaging chamber. Okay, we do a viral injection into area CA1 of the hippocampus, and then with some imaging activity in area CA1. Now, area CA1 contains place cells that respond when an animal is in a particular location in the environment, if it can remember where it is. Okay, so it's actually a fairly good readout of, if you've got good place cells, then you know where you are. It's a good readout of the quality of spatial memory. So we can use that actually as a way to probe memory function in these mice. In this particular variant, we've got a red and a green indicator. The red one is for a structural marker. In some later work, we actually use the red one to label different cell types. Okay, and then the green one is the functional one. And I think there's a movie you should be playing here on the left here of basically these are sort of the calcium, the patterns of activity you get, while a mouse is running. So in this case, the mouse is running around in a circle, and that's the pattern of activity in the brain while it occurs. Now, the other thing we can do, and this is a nice aspect of two-photon microscopy that you don't get from single-photon microscopy, because we can basically map the brain in 3D. We've got this small point for its function. Realistically, we can put it at one depth and do frame scanning to get sort of functional movies like the one I've showed over there. But in a slightly more slow timescale, we can actually just map the full three-dimensional depth, and we can map where all the amyloid plaques are. So we inject a dye called methoxyXO4 intraperitoneally in the mouse, and basically it crosses the blood-brain barrier and labels amyloid plaques in the brain for about a week or so. So now that's excited down. So we use a laser with wavelength about 920 nanometers to optimally image GCAM. Okay, this is sort of around in the 700s basically to optimally sort of excite this methoxyXO4. So we don't do it at the same time. But basically, the first few days, so we've implanted this imaging chamber. We may image these mice sort of again and again over many months. But the first few days of imaging, we basically spend mapping the brain, firstly finding where the good, sort of the nice patches of neurons are that are sort of nicely labeled, etc. We're going to be looking at, and then where all the amyloid plaques are relative to those neurons, so that we can then go back later and look at any of the results that we see. How do they depend upon the presence of amyloid plaques and the vicinity of that neuron? We can look at that neuron by neuron. So I mentioned we did this in behaving mice. So this is the old two-photon microscope. So this is the microscope, not the mesoscopes. That's what a standard one looks like. This is a little imaging, the little carbon fiber chamber that they run around in. So they're head fixed, but they're running on this chamber floating on an airbed. So they stay in one spot. Okay, their feet move, but they can sort of navigate around this sort of this maze, and they can run around either circular tracks. We do that one quite a lot because that lets us sample space quite well. We can get many trials of sort of sampling space, but we can also get them doing all sorts of other little mazes as well. And you can see here the mouse is sort of running around this sort of circular track, and you can see that there. Well, we're imaging. Okay, so
Excuse me, are you finishing?
it's no, I don't think so.
2 hour booking for this room.
I booked this room for a load of high school students. Oh, okay, well, we'll just have to do,
Sorry, I mean, I'm just a speaker.
right,
Yeah.
that's very strange, okay. Sorry.
All right, I'll just keep going.
Yeah, go for it.
Okay, so okay, this is the kind of raw images that we get out of the microscope. You can see this is sort of the green channel and the purple channel corresponding to GCAMP and methoxyXO4, the labeling the plaques. We use basically a software package which sort of uses local correlation to identify regions of interest corresponding to individual cells. So basically, if as you saw from before, you saw these sort of flashes. So basically, the pixels that are in the same cell are going to be correlated together, and the pixels that are in a different cell or out in the background are going to be uncorrelated. So you can sort of group the cells together on that basis, form these regions of interest that correspond to all of the different neurons. Then at each frame, you basically average those together in one cell. You get one of these points in the time series, and this is sort of the fluorescence. This is the calcium fluorescence, and this is just referred to the sort of the baseline. From there, we basically infer spikes. We basically we deconvolve it. So this is a calcium. So basically, neuron fires an action potential. You tend to get like a long seconds long decay of the calcium, right? You can sort of take that calcium and then go back to what kind of firing rate or probability of spiking would have produced a calcium transient like that. And so you basically effectively get the amplitudes of these kind of spikes. And that's what we plot down here. These are events, but they're events with an amplitude. Okay. Now, as I said, we do this in animals that are running around these tracks. We put a little dot down corresponding to the location where the animal was when it fired that little event, maybe corresponding to a single action potential or a small burst of action potentials. And you can see for these 5 typical cells, they're all clustered in particular locations. So these are place cells. Basically, the cells are fairly selective for place. And as you go down here, you can see this is like unwrapping the trails, unwrapping the laps, sort of just down here. And you can see they fairly robustly sort of respond to one location in the environment. So these are the place cells. Now, that's in a wild type mouse. So what happened? What's the difference between 5XFAD, these Alzheimer mice, and the wild type mice? Now, the other thing I should say is the other thing we looked at is young versus old mice. Now, in the 5XFAD model, it's a fairly early onset model, so the mice start to develop plaques by sort of 3 or 4 months of age. And by sort of 9 to 11 months of age, there's a lot of plaques in the brain, so the brains are quite affected. So our old group is a sort of median of 8 point something months, and the young group is around 3 or so. So the first thing to say, basically, it turns out that it matters whether the animal is running or resting. When we first looked at this, it looked like a bit of a mess, and then we separated it out. If the animal is staying in one spot versus the animal is running around. If the animal is staying in one spot, it turns out in the AD mice, you get this. So this is the over here. So this is this is the top one here, is the resting state here. So the purple is the Alzheimer's. Basically, there's an elevation of firing when the animal's at rest. So I think what's happening is somehow like, the synapses have been lost, but the metabolism of the cell has just been sort of boosted up a bit to compensate for that. And you end up with this sort of spontaneous hyperactivity. But when the animal's running, it's a different story. Running, you can see this is a distribution of activity here. It's shifted to the left there. So basically during running, when the animals, so you would expect that neurons should be firing more during running than when they're at rest. And the reason for that is because many of these cells, firstly, they have speed tuning, they're positive speed tuning curves. So basically they tend to they tend to fire at higher rates at higher speeds. And then also, these are place cells, and the animal is going to be running through the place fields as it goes around the track. So you should get an increase in firing. And you do get an increase in firing, but not by as much as in the wild type mice. So we see this as a sort of dynamic range, a shrinking of the dynamic range. So the baseline activity is shifted up. It can't elevate its firing rate as much to deal with like processing information, sending things in relating to, spatial memory or speed of running or anything like this. So it's not able to use that dynamic range to signal information. It's got a compressed dynamic range, and we think this is sort of what's going on. And I'll just jump to the next slide. We can measure this. We can measure how well each of these cells is doing at conveying spatial information, and therefore its spatial memory in terms of something called mutual information. So this is, we're in an engineering department, we like sort of engineering measures. This is something developed by Shannon in the 1940s. It basically tells you how much knowing the neural response reduces your uncertainty about what location the animal is in the environment. So how much it tells you about that, right? And basically, this is a cumulative. This is like over all of the cells, many thousands of cells, and you can see basically this is a cumulative plot. So towards the left is worse, right? So basically, the AD mice are much worse. The wild type mice are much better, and there are age effects as well, but I won't go into that. Okay. So that's the story with the AD mice, and there's a bunch more bits of that story that I won't go into. Where these were animals just running around in a circle. we've trained them to do little tasks where they go up to this sort of run down the center of a corridor and they turn left one time, they've got to come back around and turn right the next time, remember where they are. So they actually have to do something. And we see similar results with that as well. So it sort of bears out that, you know, if we're doing something, that there's this reduction in dynamic range. Okay, so this was just done using a standard 2-photon microscope. I wanted to talk about starting to expand the field of view to look at bigger brain areas. And this is oriented more to cortex than to hippocampus. So this is, so we built a few of these microscopes. This one, you know, the design is called Diesel 2P. That came from some of our collaborators at University of California, Santa Barbara in the US. And you know, basically, this is what you saw before. This is sort of the optical design. It's got around a 5mm field of view. So this is a few other of these pictures of the microscope, light path and everything. Now, you know, we can do these experiments sort of as before. One additional thing we're doing is monitoring the where the eye is looking and the pupil diameter as well, which we actually do through the walls of the cage. So running on. Now, then the other thing we have to do is we have to label the tissue. Okay. Now, I told you before about injecting into the hippocampus these viruses, but now we've got the whole cortex. So how can we do that? Otherwise, we might need like a dozen injections, and it might be very sort of ununiform. So the approach we've taken here is, and this is where this comes from, to use basically these blood-brain barrier crossing viruses. So this is called the CAP-B10. So this is new viral serotypes that will cross the blood-brain barrier, and so that means they can be basically effectively sort of delivered across the entire brain to label, you know, all of the brain. Together with that, we've got a strategy to label both excitatory neurons and inhibitory neurons. So the excitatory neurons sort of labeled with one virus, the inhibitory neurons with another. So we have this viral cocktail, and the inhibitory neurons we put both a red fluorescent marker and a GCAM, and it's actually GCAM-M. It's a slightly faster calcium sensor because interneurons tend to fire a bit faster than excitatory neurons. And then the excitatory ones are this GCAM-8S. So, so we can do that, and you know, these are just, you know, they're confocal images, but are basically showing that labeling works, and you can see you can get these sort of beautiful structural images of cortical circuits with that. So we're labeling it sort of quite nicely, and showing also that we can basically get the different, you know, both excitatory neurons and inhibitory neurons at the same time. So the limitation of what we were doing before was, you know, we were just imaging cells. We didn't really know, you know, which was which. Probably now mostly excitatory cells because that's what they mostly are. But it's actually very interesting to kind of know, particularly in the context of the slide I showed you earlier, where we've got sort of inhibitory compensation mechanisms involved in some of this sort of tissue response to Alzheimer's disease. Very important to know, like, the separate contributions of excitatory and inhibitory neurons, and they may help sort of tailor therapeutic mechanisms to drugs. So we can do that. We've done that, and that's what this middle slide here is doing, tail vein injection. One of the catches with that is, so far, this tends to label layer 5 cells pretty well, and layer 2, 3 cells not as well. And that's really at the limit of the depth that we can image with a microscope. So we've tried a few different approaches as well to see what works. Ventricular injection, it turns out that's good for hippocampus, but not for the cortex. And something else, which is basically just a large but very slow injection into the cortex, we found actually works quite nicely. So we can basically just inject very, very slowly and label the entire cortex with the virus, and that might in practice be better than this. This is really nice in principle, but we still have the bias effect, and also just the strength of expression is not quite as good as directly injection, because you lose expression when you go across the blood-brain barrier. Okay, so that's that microscope. Now I want to introduce you at the end to our very last microscope. So this one's actually in Suzhou in China. That's in collaboration with a group, a Chinese Academy of Science optics group, that have built this up, and they've designed this objective lens, which has got an 8mm field of view. So, and really, I think 8mm is a sort of as far as you want to go. As I said, the mouse is for mouse imaging. For larger animal imaging, bigger might still be better, but for mice, there's no point in getting anything any bigger because the curvature of the brain starts to really affect things. And it turns out making an objective lens with curvature is possible, but very, very difficult. They have done. So anyway, this is this microscope, and we can have also a sort of a curved replacement, sort of glass replacement that sort of replaces a bit of the skull over the imaging window, and then we can see. This is what we can see through it. This is done. Another aspect of this is it uses adaptive optics. That's sort of done on the fly. So you have to sort of you have some little calibrations basically, and then it sort of compensates across the field of view, so you can get good imaging everywhere in the field of view. This is a 5mm. This is a 5mm craniotomy. Still only 5mm at the moment. We'll actually bigger in a minute. But then this is an actual image, right? Right. This is the number of neurons we're able to observe simultaneously, and these are basically individual regions of interest that are a few hundred microns in diameter that we're imaging. I think they're 500 microns in diameter, simultaneously imaging. And this is the cell bodies of these are actually just interneurons. The way we're labeling this particular mouse, we're just looking at interneurons there, but we can simultaneously look at all of them across the brain there. We can use it for other things as well. So this is whole brain vascular imaging. Again, what we do here is we inject dye into the tail vein of the mouse, and then you basically image the vasculature. You can actually zoom in and because this is done in vivo, so you can zoom in and actually watch the negative images of red blood cells sort of coming past and use that to measure local blood velocity in sort of many different locations in the brain. And of course, we can use it to do functional imaging too with GCAM imaging. Now, in this case, we've used a transgenic mouse just for practical logistical reasons. So this is an old slide. Now 10,000 neurons simultaneously we can image, and you can see this is an 8 by 1mm field of view that we're imaging simultaneously at around 6 frames per second. So 6 frames per second is good enough to detect calcium signals from individual neurons and basically turn it into spike trains, something like spike trains. In the same microscope, we can basically zoom in and look at dendritic spines instead. It's a different application, but basically that just sort of shows the versatility. And yeah, so we do this again. We're doing it with in a behaving mouse. So basically, we use this behavior platform sort of across all of our experiments. This mouse is, I was talking to you before about sort of trying to run down this central corridor. Turn left one time, has to turn right the next time. There's these little servo motor activators there that sort of drop down, and we can actually have a little delay task where the mouse, you get the center point, the servo motor things drop down to stop it from going any further until a delay period, say, sort of 7 seconds or something like that, has passed. So it has to wait. Then when that time's up, the things come back up, let it proceed. And so we can use it for a sort of working memory type task, again, to probe different aspects of memory of disrupted Alzheimer's disease. And then we're doing imaging with this. So this is these are actually 10,000 neurons recorded simultaneously at 5.5 hertz. So if you zoom in on the individual sort of pixels there, you get basically these sort of time series from the individual neurons. Yeah, and that's the movie as sort of seen, sort of flashing away, 10,000 neurons all flashing away talking to each other. Yeah, similarly here, this is over a 2 by 7 millimeter region. at 6.3 hertz. So we can basically sort of image at good enough frame rates to measure individual cell activity, for around 10,000 neurons distributed across a big chunk of the cortex, across multiple sort of brain regions. Now, how do we take it further? We've actually had some thoughts on this. The trouble is, you could, for instance, if you want to get more cells, you could. This is only one plane. You could do more planes. Maybe move the objective lens down and have another plane like 20 microns below or something like this. The trouble is, you lose at least half of your frame rate by doing that, right? So you're rapidly going to get in a scenario where, now I personally think about 5 or 6 hertz is the lowest we want to go for imaging neural activity. Some people are doing it with like 2 or 3 hertz. I don't. It depends on your questions. For the kind of things we want to do, I don't think that's adequate. So we're at a bit of a stop there. But another approach we can take. So we've got. I showed you this point of light that we're the point spread function that we're scanning around in 3D. What we can do instead is to turn it into a pencil of light. Okay, so it's basically it's sort of like a lightsaber, but then scan that around. That simultaneously stimulates like at one location here, but everywhere deep, for say 150 microns deep into the brain, and then you can scan that around. The problem is you then got, say, all of the neurons that might underlie that location are giving rise to whatever excited photons you detect on your photomultiplier tube. So you've got to demix them. So it becomes a computational problem. And so we're working on that now. How do you solve that computational problem to demix that? But it looks like it's quite doable using non-negative matrix factorization type approaches. Our back of the envelope calculation says that if you've got the right mouse, nicely densely labeled, you should be able to get to 100,000 neurons simultaneously. The densely labeled is the right mouse doesn't actually exist right now, or at least it does, but there are problems with it, like it, they tend to have epileptiform activity and things. But that's an ongoing process. But the right mouse can exist, and it will soon, I'm sure. Or we can go back to the viral approaches as well, which should solve that problem too. Yeah. Okay. So, okay, and then that's just the calcium rules. The final thing I wanted to talk about is actually coming back to the hippocampus. Something we're very interested in that we can't get from calcium imaging are these signals called sharp wave ripples. So basically, sharp wave ripples are oscillations of about 100 milliseconds long of 130 to 200 hertz activity. So look like this, basically. And there's two aspects to it. There's the sharp wave bit and the ripple bit. The sharp wave bit is in one layer of area CA1, and the ripple bit is in another one, but they sort of happen together. Now, these are the most synchronous non-pathological oscillations in the brain. They happen in slow wave sleep, like non-REM sleep. And what seems to happen is you get replay of events that occur during the day in compressed time during the sharp wave ripples that are probably, and the timing of that is probably associated with the timescale of like spike timing dependent plasticity. It's basically the mechanism of memory consolidation, we think. So this becomes a very interesting thing to look at, particularly in the context of neurological disorders which have memory impairment aspects. Is this particularly disrupted? So that's something we're looking at the moment. I should say you also get them in a wake. You can get them in your wake state as well, but particularly when you're not doing something. So that could still be just a mechanism of memory consolidation. It could also be planning. You can get them in, so maybe recall for planning as well. We've these are some sharp wave ripple recordings from the lab, which are done by a local field potential electrode inserted into the hippocampus, and then we've simultaneously imaged calcium across the cortex while we're doing that, and we're trying to look for the sort of the signatures of this memory consolidation. So that's sort of ongoing work at the moment. Yeah, that's just assuming very early sort of data, showing some examples where you get this sort of sharp wave ripples and sort of aligned calcium transients in a few cells in the objects when that happens. So just to bring it finally back to this sort of precision neuroscience sort of idea that I mentioned at the start. So we're doing this work in mouse models, and we can study brainwave mechanisms of memory consolidation using our technology. We can record short wave ripples in the hippocampus while imaging across the cortex, and ask questions like how do neocortical representations reorganize as you get memory consolidation. How and in mouse models of Alzheimer's, we can then ask how does that get impaired. Now, if we want to then treat that, we've thought of ways you can go about doing that. So one way approach to that is to try and use the brain's own mechanisms of memory consolidation and amplify them. So if we can in real time detect the onset of sharp wave ripples, and then, for instance, turn on optogenetic stimulation to basically, for instance, lengthen the ripple, so you basically just get sort of more activity in that time, or maybe amplify the effect of it. We're looking at the lengthening bit at the moment. Then maybe you can use that to sort of enhance memory performance. There's some evidence from a paper from the Bajaki group from around 2019 that does work with lengthened ripples. We're working on that at the moment in my group, and we can test that in a mouse model. But if you were to do that, how would you translate it to a human? So we can't do optogenetics in human. So the idea is basically to use magnetoencephalography. So, and we've done this so far with traditional squid, these sort of magnetoencephalography things that have to be run at liquid helium temperatures. There's a new technology called OPM MEG, optically pumped magnetometer MEG, which effectively can be in this little helmet, a bit like an EEG cap, basically. And so we're looking at using that to detect the sharp wave ripples, and then the idea is that would then trigger focused ultrasound stimulation of the hippocampus to sort of excite as a sort of memory thing. This is sort of longer term vision, but the idea really is that using this sort of precision neuroscience approach, can we sort of dissect out particular mechanisms that can be then targeted for amplification, and then to sort of map that over to technologies which are usable in the human. So that's where I'd like to end it, and just sort of acknowledging a lot of people from my group who've worked on this, and thank you for listening. And I'm happy to take questions either on details of the talk or any other aspect of Neurotech.
Back to the six framework of the taking photos of the activity. What do you miss a lot? Six, like what is the actual required level to pick up all the activity?
Yeah, so basically, I mean, it depends on what matters. So basically, you're not detecting single action potentials anyway. These calcium transients, so they look sort of separate, but if you simultaneously record like with the patch pipette electrodes and the calcium signals, you can see like you'll have like a small cluster of action potentials, which gives one calcium transient. So if you care about the precise timing of spikes, you're basically lost with this technology. It's the wrong approach. We can, if you push it to the limit, you can maybe get the resolution down to about sort of 80 milliseconds. But that's pushing it to the limit, and we're pushing it to a different limit by trying to image large numbers of cells. It's probably the wrong technology if you care about precise timing of spike trends. But probably for most applications, that's not what matters so much. And you can get a reasonable estimate of like the firing rate, and we can calibrate that in various ways as well. So we can get the firing rate on a time scale of a few hundred milliseconds. So we can get a decent estimate. But yeah, so 5 hertz, 5 or 6 hertz, night course tells you what that you can do there, right? So it's yeah, that's more would be better, but I think in practice, I found that tends to be sort of good enough to get. It's diminishing returns after that for the signals, at least in areas like the hippocampus. Might be a different story. Auditory cortex. Yeah. Yes. Yeah, no.
Thank you so much for this.
Yeah. Yep.
I didn't know this great. I'm just wondering what is the ceiling in how many
Yeah. I mean, in theory, you can go more, but in practice, we don't have the laser sources, and I think there's many reasons why it would be impractical. So I think in practice 3, I'm already doing things with 3-photo microscopy. That's been a kind of a bit of an adventure. Maybe type 2 fun. The first time we actually, just before the pandemic, we built this. We borrowed a microscope, okay, and we built a pulse compressor. So you need really short pulses for 3-photo microscopy. So for 2-photo microscope, you can have maybe 100 to 150 femtosecond pulses. It's kind of okay. For 3-photo microscope, they need to be sort of 50 to 60 femtosecond, and they need to be perfectly shaped. Okay, so we built this. We basically took these pulses from this other laser we got, and we compressed them down into this sort of size, but they were a bit lopsided. Okay, so they weren't perfect. And basically, any energy which isn't going into 3-photon excitation in your tissue is going into heat. So we got it working, but one time sometimes when you're doing this microscopy, like as you go deep, as you focus deeper and deeper into tissue, you turn your laser power up, right, to be able to see deeper into tissue. Sometimes you forget to turn it back down when you go back up, and then the screen goes white. Okay, that's not what happened this time. When we did this with the 3-photon microscope, what happened is we ended up with this perfect conic ablation brain. Basically, it just went. So, you've got to be careful with it. And the other thing we found is that the lasers for this are, they're very expensive, like £300,000 laser basically for this. And they don't work reliably. It will work for a while, and then 3 weeks later, the pulses, like various things inside the laser, will go out of alignment. So nothing is our fault, but actually just drift of the components in the laser. So they're very hard to sort of keep working. There are now newer lasers which may be a little more turnkey, but it's like, with the 2-photon microscopes, you can basically, they're just plug and play. You can turn the laser on, and it kind of works. With 3-photon microscopy, it's more of a physics experiment still. Right? You're just going to keep everything working just right, and then maybe on a good day you get some data. Yeah.
So I know obviously you talked about knowing the biological elements, so mice, the humans, and also the technical. But focusing on the biological part, and I know that you said a little bit about right mice existing, the variants in humans, but what do you think your current biggest biological bottleneck is in research?
I would. Probably still the sensors. Being able to have in the right cells, with controllable density and expression. So basically, some cases you might need every cell, some cases you need sparse, right? Very high signal to noise ratio indicators, and ideally we wouldn't be imaging calcium, we'd be imaging voltage. Okay. Now the problem with voltage, there's several problems. One is that the voltage sensors are, traditionally they're about 1,000 times worse. They've actually got better over the past few years. There's Voltron and various other sort of voltage indicators. The other side of it is action potentials last a millisecond. Right? You can only count so many photons in milliseconds. We're looking at like calcium transients that might last a second. You've got a lot more counting times. And aside from the sensors, you're trying to study something shorter. Now, what we might often do is maybe not try and get those millisecond action potentials with photosynthesis, but use something which effectively has got a bit of a smoothing function. Like it takes it and maybe smears it out in something that's more like about 10 milliseconds. And that gives us a bit more time to be counting the photons, right? So that might help a bit. And then, so one of my colleagues, Amanda Faust, has been developing light field imaging for voltage sensing. Light field imaging is something where basically you image you image through a micro lens array. So basically the image gets broken up into 1,000 little images, and then you can computationally reconstruct the full image from that. But you don't just get that full image, you actually get a depth image. And you can effectively for free get depth. Okay, so this is fantastic, like magic. That the trouble, there isn't any free lunch. So basically, that spatial resolution is not, it's barely able to do cellular resolution. And they've been working on basically AI-based algorithms for, they'll do a 2-photon microscopy structural image on the same tissue, and then use that to basically they train basically an artificial neural network to basically reconstruct the higher resolution version, and that seems to be working quite nicely actually. So we're kind of getting that going, and with that, so the aim there would be able to have, and they probably still need better voltage sensors, but they're improving. But basically, kilohertz frame rate imaging or near kilohertz. You practice at 200 hertz is kind of maybe that's where they are now. Maybe you need to be closer to kilohertz with large fields of view. And they're kind of getting there. The downside of this is they can't image deep. Okay? But anyway, I guess what I'm saying here is that calcium in itself is a bit of a limiting factor. And on the biology side, there's a lot of sort of biosensor approaches that allow you to start asking them some new questions. One of them is just using voltage. Another is neurotransmitter detection, for instance. There is a glutamate sensor called Blue Sniffer, great name. There is the grab sensors. There's a group at Peking University, Yulong Li, who's developed this grab sensor approach that basically you can, not all neurotransmitters, but ones that satisfy certain sort of chemical requirements. They've basically got this method of designing these sensors for it. They've basically got a bunch of them they published for acetylcholine and all these things. So you can maybe sort of start to have sensors for all of the different neurotransmitter systems, and then look at those maybe in sort of different colors, etc. To get a more full picture of what's going on inside. So there's a lot of sort of molecular neurotechnology work from the biology side of it's advanced. Sorry, I've missed you about that.
You're talking about measuring kind of looking at shockwave ripples.
Yes.
Have you thought about or any groups within around your lab looking at, rather than looking at the actual shockwave ripples, looking at the length and not depth, so to speak, but the complexity of each shockwave ripple and what information might contain?
Yeah, we've looked at it a bit. So probably the best group in the world for this is the group of Lizette de la Prida in Spain, mostly working with mice, but they've also done some work with humans. There's actually, so they've done a lot of interesting things, doing things like neural manifold approaches, but on ripple, so you can actually like see the waveform and try and look at what it means. The problem, I think, is that the ripple itself is sort of like the envelope, and it's the wave packet where the information is, but the information itself is in the actual pattern of neurons that are activated across the brain that form that experience or memory that are then being so the ripple sort of like the average, it's like that's the local field potential signal, it's the average thing, but it's actually the spatial pattern that matters. So that's why we're trying to look at the spatial pattern. Other groups are ripple. I just want to add to that though, there's a group here at King's, a neurosurgeon, Tony Valentin, that does depth electrode implanted in patients that are waiting for epilepsy surgery. So they use these electrodes to basically localize where the seizures are in people, and then these people are then a few weeks later scheduled, when they've got it all sort of mapped out, they'll go in for resection surgery to have a small area of brain removed to prevent their epilepsy. But in those 2 weeks, basically, they record activity, and the electrodes that are in the medial temporal lobe or even in hippocampus can pick up shockwave ripples. So we got hold of some of that data from Tony. I had a student sort of working on analyzing. The trouble is, of course, these are not experimental. These are recordings designed to detect epilepsy, not to detect shockwave ripples. So it's horrendously messy data. We have managed to find shockwave ripples in it, and there are other groups who've done similar things. So there is maybe some things that one can start doing with human data. He did say basically that if we can reliably do that, we could go back in, and these patients are basically sitting there bored out of their brains for 2 weeks, and so you could actually get them to do tasks, right? So I think hopefully someone will do that, whether it's me or someone.
Just a quick follow on. About like finding the shockwave ripples. I'm not too familiar with the method. Can you tell me a bit about like how do you do that?
Yeah, so basically, at one level, okay, you can, so there's the shockwave and then there's the ripple, okay? Now, in the mouse, we probably have the advantage of being able to have a local field potential electrode, or even two, in basically in 2 locations in CA1, so we can actually pick them up separately. But you wouldn't necessarily have that in human data, for instance. So often people are mostly just detecting the ripple, right? And the ripple is basically, effectively, that's basically a sliding sort of Fourier transform that you then basically you're sort of filtering out 130 to 180 hertz, and there's enough power in that. But then you have to try and avoid false positives. So what we've done is like have that, and then have also like the sharp wave bit detection, and they've got to sort of satisfy various criteria. And it's not perfect in our hands. It's not that easy to do, but there are a number of groups doing. And we actually, in practice, we tend to use wavelet-based methods, right? Basically, wavelet-based methods that are sort of, but it's the same idea, but just sort of, rather than just use pure like old-fashioned linear filters, we use the wavelet toolboxes. Yeah.
When the muscle memory
Yep.
I was wondering what was the reason for that?
Yeah. The reason is because we were sort of trying to get it spatial working memory there, and you could make the argument. So I didn't read. There's a bunch of other slides I could have given to go in a bit more detail on that bit of the talk, but basically the mouse runs so down the central corridor. Let's say if it turns left, it comes back around and then comes back and then runs the other way, right? Now, if you have no delay, maybe what the mouse is doing, and what we think it possibly is doing, is it gets its rewards for doing a correct turn a little bit after the turn, right? So we think possibly it gets to the reward point, and then it plans through to the next reward point, right? So it's just sort of planning trajectories like that, rather than keeping in mind. So rather than it being a spatial memory task, a working memory task, where it basically keeps in mind, I'm now on a left turn, or I'm now on a right turn, and you just got to remember and hold that in mind. So having this delay period, we think if the delay period is long enough, then it becomes really making use of working memory. It turns out that also makes it hippocampal dependent. So there's some data from people basically showing if you don't have a delay, mice, the data is a little bit contested, but grant reviewers seem to believe it. If you don't have a delay, then mice without a hippocampus can do this task. Now, whether they do it the same way, I don't know, but they can do it. Whereas if you do have a delay, then what did I say? So yeah, if you do have a delay, you need the hippocampus to do it.
Does that help to transfer that to humans?
Yeah, it becomes now obviously with humans, if you've got cognitive and memory tasks in humans, they're much more sophisticated, right? it's always a mistake to try and take things from human, like tasks from humans, and try and teach mice to do them, because mice are stupid. Basically, well, they're stupid for human stuff, but it turns out they're very good at doing things in their behavioral repertoires. The idea is to come up with things that are kind of more natural things for a mouse to do, but that basically that tap into the right memory systems. And so that's the idea of sort of tapping into the working memory system in ways that are analogous to these delayed match to sample tasks that, for instance, you see in the neuropsychology literature that are kind of analogous. In fact, in those cases, they might be stimuli on the screen and they flash up and you're late and another one flashes up, but they're sort of trying to get the same kind of sort of cognitive.
We don't have any questions from the audience, so I guess I'll go for one last one to sort of
Okay.
wrap up.
Yep.
It's something to do with neurotech and
Yep,
the student
sure.
neurotech.
Yeah.
How do you think that undergraduates today, students in general,
Yeah.
undergraduates in particular, can
Yeah.
contribute to so they can get involved, given that obviously you've been working in neurotech for a long time now, long before
Yeah.
any of us were in higher education. And I guess you
Yeah.
that sort of life cycle of
Yeah.
that idea and that passion to then
Sure, yeah.
become really lucrative. So
Yeah.
where do you think there is a place in the scene for undergraduate
Yeah.
enthusiasm?
So I mean, I think the first thing I'd say is probably, I mean, there's different levels you might want to get involved. At one level, it's like, maybe going to neuroscience talks and neurotech talks, sort of, understanding what's going on. But watching something isn't the same as doing it, right? So actually, if you're really interested in it, get involved in neurotech research. And I guess there's a whole side to it that you start to appreciate by really sort of doing it and the limitations. And obviously that's different for different bits of neurotech. So that's, I mean, one thing I would say. The second is there are a lot of opportunities now in terms right at this moment, I would say, and it's not clear whether it will last in terms of like spin out companies and all this sort of startup scene. Of those, I mean, there are a huge number which are sort of low hanging fruit, of which it's basically sort of things which are there's a low entry barrier for, that there will be 1,000 companies starting to do. So basically anything based upon recording EEG signals and doing a bit. So many people do it. So think carefully what you want to do. I think there are a lot of opportunities, but you've got to have a special. There's got to be something special about it that other people can't just do. And many of those special things are things you will only get by actually getting really involved in neurotech research program. Not all of them, but yeah, you've got to have unique selling. So yeah, So I guess what I'd say is get involved in the research. Your enthusiasm for the field is encouraging to all of us, which is great. Sometimes a little bit overwhelming, almost in a sense that we've been academics operate on slightly slower timescales than students do. Yeah, so I think that's just I appreciate that point. But we've got grant timescales and all these kind of things, and you'll be doing things on a quick time frame and making your life decisions about what you're going to do next. But, let's scale in the next few years. So, yeah. Sure.
Okay.
Right, everybody sure they don't have any more questions at all?
Oh,
Okay, go on.
Let's talk about multiple apps. Like,
Yeah.
I didn't really track the journey. Like I saw the collaboration with Copperson, and I saw papers come out.
Okay. Yeah.
Those kind of things. And is the Melbourne neuroscience?
Yeah, I know them reasonably well. I met up with I've never met the CEO in person, but I've exchanged a lot of emails with him. But I've met some of the people from the company when I visited Melbourne. And yeah, it's an interesting thing. I mean, I made a similar pitch at one point as well. Basically, the idea being, our computational technology is going to be environmentally limited at some point, right? You've got those massive server farms consume huge amounts of energy, etc. And yet the brain as a computing device does so with much, orders of magnitude more efficiency, etc. So maybe we can actually take cell cultured neurons and teach them to do computational dust. Now their approach is to take flat arrays of basically sort of electrodes on arrays to do it. We had a slightly different approach, was to take organoids, so three-dimensional structured tissue, and then try and teach them about the genetics. I haven't pursued that. It's kind of a little project which sort of sat there. I may try and do it in collaboration with Brett Calgan at some point. Now, is it actually? It's pretty hard to compete with silicon. It's a nice idea as a sort of technology demonstration project. Would I be investing money in it? As a kind of, I'm not so sure. But in the longer term, maybe. But I think it's probably likely that humanity will destroy the planet and rip it to pieces in preference to doing something like that. I don't know. Let's see.
Yeah. Okay.
Sure.
All right, brilliant. Well, I just want to say thank you again
Yeah,
for coming,
sure.
and
No worries.
it's been so wonderful to hear from somebody who has been at the forefront of, I guess, academia and neurotech and this sort of scene for such a long time. So thank you for giving us your time.
Thank you.
We've all, I'm sure, thoroughly enjoyed it. So let's give him another round of applause.